and offline modes. Respected Honorable Vice Chancellor Professor Dr. Nirja Gupta, Gujarat University, Ahmedabad, though due to the urgent meeting, she is not available at present. She may join later on. Dr. Piyush Patel, Registrar, Gujarat University. He is also available at the other place for the important meeting. Here we are having guest and keynote speaker Dr. Sanjeev Kumar Singh, Professor, Molecular Modeling and CAD Lab Department of Bioinformatics, Algappa University, Karaikudi, Tamil Nadu. Dr. Abhijit Kayal and Dr. Vinod Devaraji, Senior Scientist at Schrodinger and trainers for this workshop. Professor Pranav Srivastav, sir, Professor and Head of the Chemistry Department. Convener for the workshop, Professor H.D. Patel, Professor, Chemistry Department. Professor Maisuria, sir. Dr. Divya Mishra, Dr. J.J. Maru, and all the faculty members of the other departments, participants, and my dear students. I am Dr. Dilip Vasava, Assistant Professor, Department of Chemistry, Gujarat University. Warmly welcome to the 4th hands-on training workshop on computational tools for drug discovery held on 6th and 7th March 2025. Now, I request all of you to rise at your place for university song to celebrate our university spirit, unity, excellence, and tradition. Thank you. Thank you all. Now I request and invite all the dignitaries on the stage to come forward for the lighting of the lamp. As we gathered here today, we are reminded of the significance of light in our lives. Light symbolizes knowledge, wisdom, and gratitude. It illuminates our path, dispels darkness, and brings warmth to our hearts. The lamplight is symbol of the quest for knowledge and understanding. It represents the spark that ignites our curiosity, fuels our imagination, and inspire us to learn and grow. Let us work together to spread the light of knowledge and to create a brighter future for ourselves and for generations to come. Thank you so much. Though our Honorable Vice Chancellor Madam is busy with the other urgent meeting, so we are proceeding with the next program. But before that, formally I should welcome our Honorable VC Madam. It is the immense pleasure to welcome our Honorable Vice Chancellor Dr. Nirja Madam to this workshop. We are honored to have you with us today, and we are excited to share this occasion with you. Your presence today is the testament to your commitment to excellence and your passion for education. As we gather here today, we are reminded of the significance of leadership and vision in the shaping the future of our institution. And we are grateful to have a leader like you who has demonstrated exceptional leadership and a deep commitment to the advancement of our institution. Now we are having the keynote speaker for today, Dr. Sanjeev Kumar Singh. I request Professor H.D. Patel sir to present with this small token of love, appreciation, with the bouquet flowers and memento of Statue of Unity to our keynote speaker, Dr. Sanjeev Kumar Singh, Professor, Molecular Modeling and CAD Lab Department of Bioinformatics, Algappa University, Karaikudi, Tamil Nadu. Thank you, sir, for accepting our invitation. Now I request Professor Mukesh Maisuria, sir, to present with the token of our appreciation to Dr. Abhijit Kayal, Senior Scientist at Schrodinger and trainer for this workshop. Thank you so much for accepting this invitation. I now request Dr. Divya Mishra to present a token of our appreciation to Dr. Vinod Devaraji, Senior Scientist at Schrodinger and trainer for this workshop. Thank you, sir, for accepting this invitation. Now I request Dr. Maru, sir, to present a token of our appreciation to Organizing Secretary, Professor H.D. Patel, sir, Department of Chemistry, Gujarat University. Thank you. So, before proceeding, it is my privilege to introduce our Honorable Vice Chancellor, Madam. It is my privilege to introduce Professor Dr. Nirja A. Gupta, Honorable Vice Chancellor, Gujarat University, Ahmedabad, Gujarat. Formerly, Dr. Gupta Madam successfully shouldered the responsibility as a Vice Chancellor of Sanchi University of Buddhist Indic Studies, Madhya Pradesh, India. She has also worked as a principal of R.A. Bhavan Science College, R.A. Bhavan's College, Gujarat, and has been instrumental in the development the Department of Foreign Studies in Gujarat University, Ahmedabad. Dr. Gupta's credentials include a doctorate degree in English, and she has about 36 years of academic research and administrative experiences. Her academic excellence is complemented with her proficient knowledge in 2 foreign languages, Russian and Greek, apart from 9 Indian languages such as Hindi, Sanskrit, English, Gujarati, Marathi, Assami, Urdu, Oriya, Pali, and Prakrit. She has authored more than 22 books and 60 publications in national and international conferences and seminars, and 27 students have successfully completed their PhD under her guidance. Her major areas of interest and proficiency include literature of English, Sanskrit, Pali, Gujarati, Hindi, Indian poetics, Indology, ancient Hindu Sanskriti, Pravasi and migration studies, Gandhian thought, Indian culture and faith, migratory arts and music, and she is also trained in classical dance, Kathak. She was a principal director of the team constituted on Jammu and Kashmir, a special region by Ministry of Home Affairs, and the findings were substantially used towards the abrogation of Article 370 in Jammu and Kashmir by Government of India. She has also traveled extensively in India as well as visited more than 42 countries for academic purposes. She has participated in over 400 national and international research-oriented conferences, workshops, and seminars. Some of the other key roles as a member of the Interreligious Dialogue Committee constituted by Revered Pope and spokesperson of the Citizen Amendment Act. Professor Gupta is recipient of several prestigious awards. Bharat Kirtiman Padak 2023 from World Books of Records, London. Ajka Karmavir Award 2022. National Talent Search Award 2020. Bharat Manthan Award 2020. Atal Bihari Vajpayee Rashtriya Samman for contribution in diaspora studies in 2017 by Indian Political Science Association. Shiksha Bharati Puraskar 2011 and Best Multiple President in 2001 by Lions Club District. She has been invited under Mission Education to visit the Canadian state universities, Japan, Nepal, UK, and several East Africa and Arab countries as an educational delegation from India. She has played an active role in disaster relief in Gujarat earthquake, Kashmir floods, Sri Lanka tsunami, and Uttarakhand floods. Her theme paper on G20 found a special mention by Honorable Prime Minister Sri Narendra Modi in his Mann Ki Baat on 29th January 2023 as the part of published book by her, The Mother of Democracy. Though she is not available at this point of time, she will visit during the workshop. But I should thank for her here to have as a Vice Chancellor in Gujarat University. Now, before going to the lecture, I would like to invite Professor H.D. Patel, sir, to share the glimpse of the conference, this workshop, and the Department of Chemistry. Professor H.D. Patel, sir. Namaskar. Today we are here because of our honorable, dynamic, and Iron Lady Vice Chancellor, Dr. Nirja Madam. The reason is, Madam funded rupees 18 lakhs to renew the license of this software. And when we are having license, then only we can conduct such kind of hands-on workshops. So we are very much thankful to our Honorable Vice Chancellor Madam. We are grateful that we are having this license because this license is not only useful for the chemistry people. It is useful for the various branches of sciences like bioinformatics, physics, zoology, botany, biochemistry, mathematics. In chemistry also, it is not only useful for organic chemistry, it is useful for the analytical chemistry, physical chemistry, nanotechnology, bioanalytical, and polymer chemistry. First time in 2015, we got the Schrodinger's license for the drug design, and at that time, ex-Vice Chancellor Dr. M.N. Patel sir was there. Sir told us, don't get only the software. The university will provide you the hardware support also. At that time, we purchased workstations, and then after Department of Chemistry got two major projects, and out of those, we have expanded our hardware facility. Right now, Department of Chemistry is having two workstations, and Bioinformatics Department having one GPU and super mini computer. In the field of computational chemistry, the Department of Chemistry having 100 plus research publication and more than 15 patents. Our most of the faculties are using this software. In our curriculum also, At MSc Semester 1, we are having the topic Computer in Chemistry. At MSc Semester 3, we are having Drug Design as a topic. And MSc Semester 4, we are giving opportunity to students to perform their dissertation by using computational chemistry. Why such kind of facilities are required in today's era? Today's era is known as AI-based, artificial intelligence. As we know, no one can replace the human mind, but we should know how to ready for the AI for the fast development and growth. So this type of hands-on training is important. I am as a professor, but till date, I am also learning many things. I have attended AI-based workshop at IIT BHU recently. In my career, in 2014, first time I have attended a workshop which is organized by Schrodinger at LM College of Pharmacy. After attending the workshop, the Schrodinger team has given an evaluation license that is called free of cost license, and we have carried out some work. After doing some work, we realized that this software is very much important in the field of research. And that's why we have requested, as I said, to Dr. Amin Patel sir, and Amin Patel sir started or established the computational chemistry center at our department. So I am saying that your learning attitude will enhance your knowledge and you can explore the dimension in research. So this is why you are here, why such kind of hands-on trainings are important. I have given just some glimpse of it, and I hope in 2 day sessions you will learn something. And as I said, in 2014 I have started my journey with small workshop, and today we are having full-fledged establishment in the field of computational chemistry. So we hope you will learn something and you will explore your knowledge. Thank you. Thank you, Professor H.D. Patel, sir. We will go for the keynote address, but before that, for 2 days, the trainers are going to conduct the workshop, and probably we may not able to listen them for the next 2 days, some of the participants. So before going to the keynote address, I would like to introduce the trainers for this 2 days workshop. First one is the Dr. Vinod Devaraji, Senior Scientist. Dr. Vinod is the distinguished scientist with nearly 14 years of experience in computer-aided drug discovery (CAD), contributing to advancement across various therapeutic areas. He holds a PhD in biotechnology covering diabetes research from Vellore Institute of Technology, where he integrated computational drug design, herbal therapeutics, and formulation studies to identify novel therapeutic strategies. His academic credentials include an MPharm from Madras Medical College and a bachelor's degree from Padmavati College of Pharmacy. A recognized thought leader and speaker, he has delivered numerous talks and workshops across the Asia Pacific region, focusing on the emerging scientific technologies and their role in addressing drug discovery challenges. His current research explores the application of computational formulation, artificial intelligence, machine learning, and physics-based molecular modeling to accelerate drug discovery and optimize lead identification. With around 40 publications in leading international journals, he has made significant contribution to the field, spanning diverse computational techniques and drugs design methodologies. Currently, he serves as a senior scientist at Schrodinger, where he provides global scientific consultancy and collaborators with the top academic and industry researchers to drive innovation in computational drug discovery. Welcome, sir. Another trainer for today's session is Dr. Abhijit Kayal, senior scientist. Dr. Abhijit Kayal working as a senior scientist too at Schrodinger. Before joining the Schrodinger, he was a part of with several biotech and pharma companies as a scientist. He received his PhD in computational chemistry from IIT Kanpur in 2017 in the field of theoretical chemistry. Following this, he pursued a postdoctoral position at Jülich Research Center in Germany, focusing on QM and MM stimulation of ion channels. The expertise are in ligand and structural-based drug design, molecular dynamics simulation, QM, MM-based simulation, and FAP calculation. Welcome, sir. Now I invite Professor H.D. Patel to introduce our keynote speaker, Dr. Sanjeev Kumar Singh, Professor, Molecular Modeling and CAD Lab, Department of Bioinformatics, Algappa University, Karaikudi, Tamil Nadu. Again, namaskar. Professor Sanjeev Kumar Singh is the professor at Molecular Modeling and CAD Lab, Department of Bioinformatics, Algappa University, Karaikudi, Tamil Nadu. Sir, basically from Lucknow. Sir has completed various degrees. Sir is having DSc in Bioinformatics, PhD in Applied Chemistry, professors in Bioinformatics, Algappa University. Before that, sir worked with Center for Biomedical Research Center, SGPGI, Lucknow, which is equivalent to AIMS, as a director and dean and professor. In the research contribution, sir is having 232 publications, 42 book chapters, and 10 projects completed by sir, and 2 projects are ongoing. Sir received various awards and honors. Sir's name in world's top 2% scientist, which is declared by Stanford University 2022-24. Sir is having INSA Teachers Award 2022 by Indian National Science Academy, Fellow of Royal Society of Chemistry and Biology. Sir received Outstanding Researcher Award. In professional roles, sir worked as a research guide. Many students are guided by him. Sir organized various conferences and delivered 71 invited lectures. His key positions are member planning board of Algappa University, director and coordinator of RUSA and RUSA 2.0, MHRD, Government of India, and received 100 crore rupees as a fund. So with this short information, or we can say introduction, Now I am requesting sir to give his talk. So we will get the information how drug discovery is important. And then after Dilip sir will proceed.
Which mic I have to use? This mic or this mic? Both are there. Both are okay. Oh, this is how you can switch off this one. I can use this one. My phone is there. Phone, mobile phone. This is not working. Why is the slide? It's working? Okay. Can you remove? It's stable. Maybe you can remove. Thank you. Right. So now it's okay. Why is it audible to everyone? Right. So, like whenever we talk about the drug discovery or computational drug discovery or even any disease, so this is the geographical location, like how like entire world is suffering with the n number of diseases, n number of viral infection, bacterial infection, or n number of infection. Right. Recently, we have come across in the year 2019 and 20 with the one huge pandemic called COVID-19. Right. We are not at all mentally prepared for that. And this was not happened the first time. In earlier days also, we have our generation has faced some other challenges, right? So if you look into it, like polio, now it is completely eradicated with the efforts of scientists and researchers. We have now good vaccine for the polio. We have the medicine for the polio, and there is no polio cases now available. Anthrax, Ebola, measles, HPV, very common nowadays, right? If you see here, Guillain-Barré syndrome, last month we are listening a lot about it. Okay, this is re-emerging. So what I mean to say over here is that there are thousands of infections, thousands of diseases. So our responsibility, the researcher, those who are working in the area of drug discovery is enhancing every day. Our responsibility is enhancing every day. What we have to do, we have to come up with a molecule which is having the good potency and good value for the population. Coming to the next slide, if you see this is a viral spread over the world. Out of this, there are few are newly emerging viruses like H1N1, H5N1, influenza, SARS. This is initially for the bat, but now it is for the human. There are so many there, those who are re-emerging and resurging is like Nipah virus. We have come across a number of things, H1N1, Nipah, swine flu, and most common is like dengue and chikungunya, which everybody knows, right? We still do not have a very effective medicine for the dengue and chikungunya, right? And you will be surprised to know that the kind of the casualty happen, the kind of people die because of the dengue and chikungunya is much more higher than the people died because of the COVID-19, right? And every day few casualties are there because of the dengue, right? So the workload of the viruses are also very high, and there are so many viruses like Zika, Ebola, which is not very common in India. This is more for the African population. And whenever any strain comes to India also, it is not very active, right? But yes, we have to bother about that. We have to bother about new infection. Coming to the, and in my today's lecture, I will mainly concentrate on the HIV. Why HIV? Because as we know, uh, among the viral infection, the HIV is one of the oldest infection. From 1981, it was introduced. And from 1981, and this is 2025, we do not have any vaccine for the HIV, right? Almost 45 years, we do not have any vaccine for the HIV. But luckily we got for the COVID, we got the vaccine, Covaxin, Covishield we got, and we are able to save lots of population. So not only the HIV, but there are lots of viral infections are there for which we do not have vaccine. We are struggling because of the chikungunya and dengue, but we are not able to develop the vaccine for that, right? So along with that, this is very interesting that one notorious insect which is creating more problem for the humankind is what? Mosquito. Each and every individual bother about mosquito. Even yesterday when I entered here in the guest house, I asked Professor Hitesh whether the mosquito is here or not, whether my target insect is available here or not. Right. So see, you see in the list, Zika, malaria, yellow fever, West Nile, dengue, Japanese encephalitis. This is all because of the one which is mosquito. Right. And this is the geographical distribution around the world because of this insect. And WHO has already declared this is one of the very notorious insect. And in future also we should be ready for that. Like we can have more infection because of this mosquito. And uh, one more thing which I can tell that initially when we COVID-19 come across in the picture, initially we have used the medicine for the malaria. When Indian Council of Medical Research has released the first advisory, the chloroquine is the first anti-malarial drug which was introduced for the COVID patient. And slowly every way, because that time the severity was very high and we do not have any options available for that. So we have to come up with some molecule which can get some at least temporary relief for the COVID patient. So we have tried anti-HIV drugs, we have tried anti-malarial drug, we have tried like the drug developed for the Ebola and Zika, or like lots of antivirals we have tried, right? So this is all detail about my lab, like where we all are working. Our main group is working on the HIV, and we also work on the Zika virus, chikungunya, dengue, and last 4 or 5 years we started working on the SARS-CoV also. And along with that, we also working on the two viruses, which is HPV, papilloma virus, and HTLV. And what these viruses are doing, these are causing cancer. These two viruses are causing cancer. And apart from that, the two students are working on the cyclin-dependent kinase and hostile transferase. And two students are working on the OREI1 and STI1, like neurological disorder. And three students are working on the antibacterial targets, right? And our work is mainly on the molecular mechanics, quantum mechanics, computer drug design, protein-protein interaction, and GS data analysis, and so on. For more detail, you can just go to the Sanjeev's Lab website. So coming to the, like our today's topic on the HIV. So if you see over here from 1981, the AIDS outbreak was reported. And after almost 5 years, the AZT, azithromycin, this particular molecule I have used in some of the preliminary data for my study. And the year 1991, the first protease inhibitor, like I will tell what is this protease integrase. And you see that this timeline in 2006, almost after 25 years, the raltegravir, which is a very important molecule, which was introduced in 2007. And after that, this dolutegravir was introduced in 2011. And this process is still on. Still, we are continuing with developing more novel molecules for the HIV. Right, this is the summary of the global HIV epidemic of 2019 data. People living with the HIV is around 38 million people is still living with the HIV. 1.7 million people are newly infected in the year 2019 with HIV. Right, this is a casualty because of the HIV in the year 2019. Around 7 lakh people died because of that, right? So normally what is happening, the people are not dying because of HIV. People are dying because of the co-infection which has happened. What is happening in the HIV? HIV is spoiling our defense mechanism. It is spoiling our lymphocyte cells or CD4 cells. And when the body defense mechanism was completely spoiled, then the particular person is not able to fight against any kind of infection which is happening. Either it is a small, simple cold also, right? So people are dying because of the co-infections which has happened after the HIV. These are data about Indian context. So you see, like before going into detail about the drug discovery, like how the HIV virus is really interacting inside the body. We have a CD4 cells or lymphocyte cell, which is known for the immunity of the body, right? And all the viral infection all the viral infection, any kind of, either it is a dengue, chikungunya, HIV, or whatever. Everything is, all this infection is depend upon our immunity. And this immunity word we came across while during the COVID pandemic. Like if your immunity is very high, you are very strong, you are strong enough, you are not able to, no casualty will be happen, and you are able to fight against the infection very easily. Like, so people are working on the, to enhance the immunity by this, lots of Ayurveda, lots of homeopathic medicines, like, okay, and all this vitamin C, right? So you see here, this is a HIV virus. It is going inside the HIV capsid, going inside the protein shell, and the target is the T cell or CD4 cells, which is responsible for the immunity of the body. After that, this HIV capsid moves towards the nucleus of the T cells. After that, different RNAs begins leaving the nucleus of the T cells for the replication. And this is RT is one of the enzyme to generate the complementary DNA. And this is a protease processing, like virus is ready for the maturation process. And this is fission occurs in the CD4 cells, and this virus is coming out, maturing, and then ready for the new infection. And this process is continued till the entire body's defense mechanism was not completely spoiled, right? When all the T cells was not completely occupied with the HIV virus. These are the mechanism of the replication, like where actually we are concentrating. So this is, we are concentrating on the fusion inhibitors, means we are trying to find out the inhibitor which blocks the HIV capsid proteins so that the HIV virus is not able to interact with the CD4 cells, right? This is the one thing. Second thing that we are working on the reverse transcription, like to stop the replication process. We are also working on the integration process. This particular integration and the cofactor interaction, I will explain in detail in today's lecture. And after that, we are also working on the protease inhibitors, like after, because this particular virus before maturation is not ready for the infection. So here we are working on the protease inhibitors. When virus is going towards the maturation, before that itself, we should suppress the virus so that it cannot able to interact with the lymphocyte cells. And also recently from last 4 to 5 years, we are working on the latency, which is one of the very main cause, like why we are not come out from the HIV problem in last 45 years. So I will explain about this. So these are the approved medicine for the HIV in this last 25 to 30 years. Some of the molecules are protease inhibitors, some are the integrase inhibitors, some are the cofactor inhibitors, some are the fusion inhibitors, right? So before going into the detail about the drug discovery, let's understand what actually the drug discovery process is, how it is complicated, and what exactly we are doing to find out a novel molecule, right? Where is our role involved? Where is your role involved? And where the other peoples are involved in the discovery process, right? So first thing is to identify the disease, which takes again several 2 to 3 years to identify the disease. Suppose sometimes we are not able to understand the real cause, where the disease really originates, what is the responsible protein in the body which is causing this particular disease, right? Suppose if you talk about the recent in the COVID-19, we are working on the multiple targets, whether it is the RNA dependent RNA polymerase, whether this is the other proteins, so identifying the novel protein, identifying the novel biomarker, identifying the novel gene which is responsible for the disease is very important, right? Nowadays, you may have heard about those who are the biotechnology people. Uh, we are we have identified the very novel biomarkers for every disease. Like for prostate cancer, some particular gene or biomarker is there. So doctor is suggesting that after age 40 or 45, you have to go for this all this testing and biomarker analysis so that you can understand that whether that particular gene is existing in your body or not, so that you can take precautions well in advance, right? So identifying the protein is very, very important. And first, when identify the protein, after that, we have to isolate the protein which is responsible for that, which again takes 2 to 5 years. Why I'm telling this is because this is a drug discovery and development and designing process is such a complicated process, right? So isolation of the protein is a bit complicated. And after that, once we identify the novel protein responsible for particular disease, we have to go for the finding out the effective uh, molecule against that particular protein, which again takes 2 to 5 years. And after that, we have to go for the preclinical testing, which is on the small animals, rats, guinea pigs, and rabbit, and then monkey, and then human. And after that, we have to go for the scale-up process. What is the scale-up process? Directly, indirectly, everybody is aware about that. The quantity of the drug molecule which is required for the inhibition, right? Whenever you go to doctor, he is suggesting you know that this particular medicine required in 500 mg, this 1000 mg, 100 mg, or whatever, right? So here in this particular area, we decide that how much quantity of the drug molecule is required for the inhibition, right? Like inhibitory concentration IC or minimum inhibitory concentration MIC or effective dose ED or lethal dose LD, right? And after that, we have to go for the formulation where we have really in the pharmaceutical development, this is again a very important area where we decide the molecule, whether we want that molecule in the form of tablet, capsule, injection, and in injection also it is intravenous or muscular or whatever, right? And after that, we file the molecule for the IND approval. This is nothing but the identification of new drug. And after the identification of new drug, we got the approval for the going for the human clinical trials, right? And this is one of the very complicated process in the discovery, which take 2 to 10 years. And after that, this is the end filing for the NDA, which is a new drug administration. And after NDA, this is approval for the FDA. So overall, if you see that, so one particular molecule, one drug molecule to bring it to the market takes around 12 to 15 years, right? And this is a drug discovery pipeline. We start with the large data set and finally sometimes getting one or two molecules. Or in some cases, we never get any molecule or still we are working on that. We start with the 10,000 or lakhs of molecule initially, but sometimes we get no molecule. And as an average, around 800 to 1200 million US dollar, which is required, and then 12 to 15 years time is required. Now, the impact of technology in the development process. Where is our role? As a student, as a researcher, as an academician, as a scientist, what we can do in every area, right? What is our contribution, right? So, biologists can work mainly on this part. Oh, and then isolation of the protein. We have several methods of isolating the protein for solving their structure. We have one very important technique called as X-ray crystallography by which we solve the protein structure. If X-ray is not available, then with the computational method, the team Schrodinger will also explain you about the protein modeling or homology modeling, right? How to get the three-dimensional protein structure if the structure is not available in the protein data bank, right? Then we apply the genomics, proteomics, and pharmacogenomics, where we can talk about the personalized target. Nowadays, people have heard about the personalized medicine. What do you mean by personalized medicine? Nothing but like some of the drug molecule which is common or which is important and useful for one population may not be useful for the other population. Why? Because the genetics is different, the genes are different, and the mode of action of the drug is different. So now whenever we go for the clinical trials, we take analysis of the pharmacogenomics, we take the people from the variety of having the different genetics or different genomes, and then we keep in the consideration and then developing a small molecule. After that, high throughput screening. This is unlike earlier days when we have to first synthesize the molecule and then go to the biologist to check the molecule. Now we can first evaluate lakhs of molecule on the screen, check the interaction with the protein, check the interaction with the DNA and other biomolecules, and then we suggest that okay, this set of few molecules are only the important one. We can take it further, only the few molecules, not the all the molecules. Right, virtual screening, again the same thing. We can screen lakhs of molecules on the screen. Combinatorial chemistry, we can generate rapidly producing the vast number of compound from the good molecule, right? with the changing the side chain, with the changing the core group. All those things will be explained by the team Schrodinger in the practical session. And then the molecular modeling, right? So here, I will take just a little bit pause and just tell you that, see, now if you talk about the drug and the disease, there are thousands of drugs are already available. There are thousands of drug is available with us, but still we are working on that. Suppose if you talk about the cancer, we have a drug for all type of cancers. That's why the medication is going on in the hospitals, right? From India to US, we have a very good number of molecules or drug molecules, efficient molecules for the chemotherapy or radiation therapy or whatever. It is available with us, right? But even then, what is the need of developing the doing the drug discovery on the cancer? Why it is required? Because every time we are working on the efficacy, accuracy, zero side effects, less side effects, right? Because what is the problem, main problem with all the drugs? That every foreign molecule, every drug molecule which we are consuming is having side effects, right? So that is why whenever we are doing the drug discovery, we have the ADME property analysis. Like when we evaluate the drug molecule, it should have the strong absorption, distribution, metabolism, but it should have the very strong excretion level also. After the performance done in the body, the molecule should go out from the body, right? So there are thousands of drugs are lists are available on the website where we know that these particular drug molecules were removed from the market. These drugs are having huge side effects. Some drugs are creating high BP problem, right? Like one very common example, Vic section 500. Vic section 500 was banned molecule. Aspirin, banned molecule, right? So few places still it is in use because we do not have any other substitute, but still hundreds of drugs are removed from the market after having the long research, 15, 20 years of the research on that molecule. Because why? Even then, so you should understand that sometimes even the 15, 20 years is not enough to evaluate the drug molecule, to check efficiently, right? So we are doing the molecular modeling, means we are keep refining the existing molecule, we are keep developing the molecule which is having the zero side effects or very, very less side effects, right? And nowadays we are also talking about the in vitro in silico ADME model, which can replace the animal testing, right? Animal testing for the drug molecule cannot be removed completely, but yes, we can reduce, right? In place of sacrificing 500 rats for the testing of the one drug molecule, we can sacrifice only 50 rats, right? So we can replace the animal testing by what way? We can take the in silico models of the ADME and we can check that, okay, if the computational value of this particular molecule in this range, only it is an active molecule, remaining properties can may not be good for the effectiveness, right? Right. So I'm coming now on the HIV integrase. As I told in the beginning, that we are working on the attachment inhibitors of the HIV. Like we can stop the interaction of the HIV to the human host cell, CD4 cell, or we can work on the reverse transcriptase inhibitor, or we can work on the stop the integration process, or we can stop the maturation process of the HIV, or we can stop the latency, right? So I will take a little bit about to you to the integrase process. So this is the integrase timelines from AIDS outbreak. After that, it is 1993. This is a diketo compound was 2000 diketo acid was reported, and after that, in the 7 and 12 and 13, these are the three molecules, raltegravir, alvedgravir, and dolutegravir. These three molecules, even still, the kind of the integrase molecule which we are getting now, also having the same kind of a structure or same kind of mode of action, which earlier in 2007, the raltegravir was developed, right? So coming to the strategies, see, either we have to stop the DNA binding, the integrase and DNA binding, or we have to stop the oligomerization, like converting from the monomer to dimer, tri and tetramer. We have to stop it, or we have to stop the protein-protein interaction. For this particular thing also, I have a couple of slides to explain about how the protein-protein interaction blocker can have the very good drug molecule. And strand transfer inhibitors means we have to stop the virus to interact with the host cell. This I will explain in detail. And another one is the allosteric site. Other than the active site, right? Every protein is having the active site, which is like most conserved region or most important region where the drug molecule is binding. And there is one more site, is allosteric site, which also sometimes play important role in the developing the drug molecule. Right. This is a metal chelation mechanism, or in the other way, we can say that strand transfer molecule design. So this is a HIV integrase. This is an integrase protein virus interacting with the host DNA through this phosphate. And the most important thing, what is involved over here is the two metal atoms. Like two magnesium ion is important to interact with the host DNA. So what we have to do in the strand transfer inhibitors or integrase inhibitor, we have to the uh, block this magnesium ion with the inhibitor, with the drug molecule, right? And when the inhibitor is blocking this interaction of this magnesium, it is not able to interact with the host DNA, right? This is a structural domain of the HIV integrase. This is a catalytic core domain, which is chelating with the magnesium ions, and this is a N-terminal domain responsible for the binding to the zinc multimerization, and this is a C-terminal domain, which is non-specific DNA binding. So this is a structural domain of HIV integrase. Again, back to the integrase resistant transfer inhibitor, which I told, like 2007, we have developed one molecule. In 11, we have developed one molecule. In 13, we have developed one molecule. In the year 2019 itself, we have developed another molecule, but all are having this 3O, the triad O common in all the molecules. So this particular region is responsible for the metal chelation. You see, this is a triad O, and on the other hand side, this is fluoro or chlorobenzene, which is responsible for the interaction. So I have some case study on these molecules. In the first work, what we have done, we have taken this N-methylpyrimidones, 50 compounds, and we have developed the pharmacophore. The pharmacophore explanation and the tutorial will be handled by the Schrodinger team today. I hope, Abhijit, they're going to handle the pharmacophore, right? Okay. So that time you will understand more how exactly we are performing the pharmacophoric study, which is the most common and the important reason on a drug molecule responsible for the drug's activity, right? Like if the drug is having around 50 atoms, then all 50 atoms are not responsible for the activity. Only the 4 or 5 atoms or the functional group, 1 or 2 functional groups only responsible for the activity. So we have developed in the first study the N-methylpyrimidones pharmacophore, and we have developed the 3 pharmacophore. And based on this R square value and the Q square value, we have, you see, this is a like 2 acceptor, 2 donor, and 1 aromatic region, which is controlling the activity of N-methylpyrimidone. And we have further done the QSAR, which is a quantitative structure activity relationship. Like every structure is having some activity, right? This is, I think that also will be handled by the Schrodinger team. And here, all the 50 molecules we have distributed into 2 sets, test and training, right? And you see what we are doing in the QSAR model, that we are predicting the theoretical activity of a molecule for which the experimental activity is already known, right? So we are developing a theoretical model, a computational model in such a way that that model should give you the accurate or very, very close value uh, in compared to the experimental value. So you can see here, all the molecules are having more or less very close experimental and the theoretical value, right? And this is nothing but the contour maps around the most active and the least active molecule. And this red cube says that which particular portion to be removed from the molecule, like which is not contributing in the drug's activity. And the blue region says that which particular region or environment should enhance in the drug molecule, right? So this is called as a favorable and unfavorable region, right? So what we are doing in the QSR, we have a multiple checkpoints. One thing is that R is square for the training set, Q is square, and these values tells that how accurate our model is, right? If the model, this is close to 0.9, the model is good. If it is away from the 0.9, the model is not good, right? This, if the red portion is very high, it means the molecule is not favorable. If the blue tubes are very high, it means molecule is favorable. And this kind of molecule or environment we need in the future molecule, right? Coming to the other study, which is based on the structure, this is DNA, viral DNA. This is the integrase domain, which I have explained. 3 domains are there. And what we have done here, that when we have done this study, that time the actual HIV integrase was not available. So we have used the surrogate model of the HIV integrase, the prototype foamivirus integrase we have used. And this is a comparison between the foamivirus integrase and HIV integrase, with very high similarities there. And as I explained that for the all 3 molecules, raltegravir, elvitegravir, dolutegravir, the triad O is very common. And fluoro and chlorobenzene on the other hand side is important. So if you see the NMP1 also, this is a triad O, either NMP1 or NMP2 we have superimposed over here. And this only triad O is interacting with the magnesium ion and controlling the methyl mechanism and controlling the activity of a drug molecule. And here we have compared the foamivirus integrase and HIV just to check that whether the exact amino acid is forming over there or some different amino acid, right? And this interaction of the triad O with the protein, we have checked through the long simulation, and throughout the simulation, we can understand that whether this particular interaction of the triad O and the metal atom is continuing, or after some time this interaction is breaking. So in the simulation, we have checked that. And this particular molecular dynamics simulation also will be handled by team, and I think in another 1 or 2 days of this your workshop. Coming to the another study which we have done on the integrase, and here what we have done, we have combinedly a ligand based and a structure based study we have done. Ligand and the structure is nothing but whenever we have the involvement of the protein structure, whenever we have the involvement of the biological macromolecule, DNA, RNA or protein in our study, it is called as a structure based study. And when we are not requiring any structure, protein or any macromolecule in the study, we are calling it as a ligand based study, right? So this study was done only on the small molecule, and this is along with the protein. And we have found some new molecules from the database, and these molecules were equally active. Why it is active? Because all are having the triad O in such a position which is interacting with the both the magnesium ion over here, right? Now coming to my next study, which I told you about the protein-protein interaction blocker, right? Whenever we talk about the HIV integrase, then the our new or like you may have heard about artificial intelligence and machine learning and understanding the very accurate and minute interaction to control the very minute interaction. For controlling any minute interaction, first we have to understand that what interaction we have to really looking for. What kind of interaction actually we have to block, right? So coming into this slide, you can understand here, this is HIV virus. This is a cytoplasm of CD4 cells. And this is the P-integration complex. And here the LGF is there, one cofactor, right? Those who are biologists can understand very well the role and importance of the cofactor. There are so many cofactors. The transmembrane SR2 is another cofactor. LGF is another cofactor. So what we have done, we have concentrated on the LGF. And this is interacting with the DNA and the integrase. And here this is coming inside the nucleus, right? So what we have done over here, we have find out that if we can understand the role of LGF and the integrase, and suppose if we can stop the interaction of the cofactor and integrase, we are able to develop a very novel molecule. in the year 2015, when we have started our study on the LegGF, one of the cofactor, it was just a second or third study in the world, like which who has started working on the LegGF, because it was not well established that whether concentrating on the LegGF is important or not. But now we have hundreds of papers, and it was proven to be the important one, right? So what we have done, this is a LegGF binding site, and this is a like HIV integrase protein, and LegGF is binding at the two sides of the integrase. This is one side, and this is the second side. And you see here, this is the interaction of the LegGF and the integrase, and these are the key amino acids: lysine, isoleucine, aspartic acid, phenylalanine, and valine, interacting with the threonine, tryptophan, glutamic acid, and histidine of the LegGF. So what we have to do? We have to stop the interaction of these amino acids to control the, to develop the potential integrase inhibitor, right? You see here, this is the, like how is it interacting. And in one of the study, what we have done, we have developed one molecule, we have designed one molecule, which is exactly working on the cleft region where the LegGF is interacting with the integrase. And when this molecule is interacting with the integrase, the LegGF is not able to bind effectively with the integrase, right? So this is a kind of a protein-protein interaction blockers. So you see, this is another study which we have done on the protein-protein interaction blockers. Right? This is molecules, design molecules exactly working on the cleft region where the LegGF and the integrase are binding. And this is the other homolumo and the MESP study. The detail about the homolumo again also will be handled by the team Schrodinger. Here, why we are doing the MESP, to understand the exact environment around the molecule. Whether this green stands for the neutral region, the red stands for the negative region, blue stands for the positive region. To understand that the exact molecular property of a molecule, like what is the requirement of the molecule, which kind of molecule we are really looking for to be an effective inhibitor, right? And here we have understanding about the nucleophilic and the electrophilic sites of the most important molecules. Right. So I explained you about the integrase. I have explained about you the protein-protein interaction blockers. Quickly, I will just take you to the other work on the reverse transcriptase inhibitor. This is the one work which we have done only on the small molecule for the reverse transcriptase. What we have done over here is that we have taken the known molecule AZT, azothiamidine. This is a clinically known molecule for the HIV, which is commonly in use. And what we have done, we have replaced at 3' position, we have replaced the N3 and we have attached the NO2 over there. We have tried multiple functional groups, NH2, NO2, N3 or whatever. And we found that the nitronucleoside, NO2, is having a very good efficiency because it is not changing the contour maps and very effectively interacting with the active site of the reverse transcriptase. Continuing to the previous work, what we have done, we have tried to find out the exact correlation between the binding affinity, binding energy, interaction energy, and IC50 value. The IC50 value which comes from the experiments, and the binding energy and the interaction energy which comes from the theoretical study. So in this graph, we have exactly plotted between the IC50 value and the interaction energy. And we found that if the molecule is having a kind of some threshold interaction energy, then only the molecule will go for the competitive inhibition, right? So this was initially I was having some kind of criticism between the directly correlation between the potency and the interaction energy, but we have like concluded there are some 12 kilocalorie per mole interaction energy as required for a molecule to go for the competitive inhibition. So all the molecule which was the effective RT inhibitors are having the interaction energy 12 kilocalorie and high. Right. So here you see this is the AZT interaction energy which is clinically proven, and this is we have tried with the NO2 over here. This is minus 21.30 kilocalorie. Now this is the same kind of a study on the non-nucleoside inhibitors of HIV-RT. Yes. Now I am coming to the very interesting part of the HIV, right? Last 45 years, hundreds of molecules, thousands of molecules, and continuous consumption of the drug molecule for the HIV. But still, the person, those who have to consume the medicine for the HIV, have to continue throughout the life. Whenever they are breaking or having any kind of irregularity in taking the medicine, the co-infection will occur and something happen. Why this is happening, right? You see here, this is the level of detection, like when the, when the HIV virus is moving in a blood stream, then we are able to identify it by our test, right? There are so many drugs are there, combined antiretroviral therapy, which is a very effective therapy, hard therapy, very effective therapy, is still going on, which is able to detect the HIV in a body up to this level. But there is a beyond this level. Even we cannot detect it. Even the medicine also not able to detect that interaction. So what is happening here? Out of 1,000 virus, one particular virus is going for the reservoir. One virus is going for the latency reservoir, going on the resting stage, right? And we have to identify that particular virus. And that is why this particular disease is not able to cure. Whenever the person is consuming the medicine, and like combination therapy. Have you heard about combination therapy? How many of you have heard about combination therapy? Only few people knows about the combination therapy, not all. Combination therapy is nothing but because the virus is developing the resistance, so we have to chase the virus, to confuse the virus, we have to change the medicine every second week or every third week, right? Sometimes protease inhibitors, sometimes integrase inhibitors, sometimes RT inhibitors, sometimes LRA or whatever, right? Even in the protease inhibitor also, one time this medicine, one time other medicine, right? So virus is not able to develop the resistance against particular infection. So that is what we are calling it a combination therapy, right? So what we are doing over here, so we have to identify the what is the cause of this latency. Like one virus is going for the reservoir. So any CRT therapy, whatever the kind of a medication is going on, that is not coming and interacting with that particular virus, which is going for the reservoir, right? So now we have start working on the, we have to work on the particular virus, and we should not allow any particular, right? So we should not allow any particular virus. He has given me the one slip that I can continue for another 50 minutes, right? 5 minutes. Okay. So we should not allow any particular virus to go to the reservoir. So what we want every virus to come in contact with the drug which we are giving to the patient. So for that, what we have to enhance the replication. And here is a contradiction. See, we have to stop the replication of the virus, and on the other hand side, we are telling that we have to enhance the replication of the virus. So what is correct? So when we have got one project from the DST along with Professor Speck in the Switzerland, because he only introduced to me about the latency problem, and we have discussed that why we are triggering the replication process. Then we have studied on that. Once we will enhance the replication process of the virus in the body, then only the particular virus will come in contact with the drugs which is we are giving to the patient, and then only it will not go to the reservoir. If we are not giving the latency reversal agent, then definitely the one particular virus goes to the reservoir, and that is why from last 45 years, person is not able to stop the medicine, right? Person has to continue with the medicine. So we have worked on this concept also, and like we have to give the LRA, so that like shock and kill strategy, and then we have to give the antiretroviral therapy. So LRA along with the antiviral agents, and both it will work for that. So we have done one QSR study on this, and we have developed a good model, and these are the final model molecule. So on the brief, this is initial work on the strand transfer inhibitor or HIV integrase. This is on the protein-protein interaction of the LGF and the protein blockers. And this is on the reverse transcriptase. This is on the protease. and this is on the latency reversal agent, right? So nowadays we are working more because now we have a very effective molecule for the HIV, and we have to work on the latency. We should not allow the virus to go for the reservoir, right? So these are my collaborators, Professor Anthony for antibacterial work, Professor Indra for the antiviral work, and Professor Banerjee also for the antiviral. And these are my Indian collaborators. These are my research project, those who have fund uh, money for doing all this work. Currently, I'm having these two big projects from the DBT. And here in one work, I'm working on the only on the latency, and on the other work, we work on the antibacterial targets, right? So these are my research group. And these four people, Dr. Reddy, he has first worked on the LegGF. Now he's a faculty at Bobby State University in USA. He is Umesh. He is also faculty in Korea right now. And this is Dr. Ishwar. And Akhil is doing on the latency. Again, now he still is working on this topic. And Aarti and Ruba is working on the HPV and HTLV respectively. Arun is working on the dengue virus. And Sunil is a postdoc in the USA, in University of Michigan, and he's having own startup at Bangalore. And this Salvaraj, he was working earlier in the FDA USA, and he's having the own company at Indore. She's working on the bacterial target. These three people are working on the NGS, whole genome sequencing data, and these two people are working on the neurological disorder. Thank you very much. This is having, you may be aware about the BIS society. Everybody has come across. So we are running one society, and for the student, we have a very meager amount to become a member, where we have all the drug discovery and the faculty and scientists are at one platform. If you become a member, you can interact with everyone anytime, right? So if you are not a member, try to avail the membership under this society. Thank you very much. I have exactly taken 5 minutes. I have taken 5 minutes only.
So it was really eye-catching and mind-blowing lecture delivered by Dr. Sanjeev Kumar Singh. And he is the person who has drawn more than 100 crore rupees for the research. And a lot of outputs are there from that grant and research. Also, everybody was shivering with the HIV because he has opened the world of the HIV and world of the research also, because a lot of technicalities were there and technical words were utilized. But one thing is very sure, that this workshop is connecting to this. For next 2 days, today and tomorrow, those who are participating in this workshop, they will learn such kind of software to develop the interaction studies. So thank you so much, sir. If you are having any questions, sir will be around. Later on, you can discuss with him and have the conversation, because he is very experienced person. So definitely you should take the benefits from his knowledge. But due to the short period of time, we are proceeding now, sir. So thank you so much, sir. Now I would like to proceed that this workshop is designed to provide you with practical, hands-on experience and expert guidance to enhance your skill and knowledge. You will have the opportunity to engage in interactive sessions, group discussions, and hands-on activities. Our expert trainers will share their expertise and provide personalized feedback to ensure you get the most out of this workshop. We are confident that you will leave this workshop with new insights, skills, and connection. Those who are participated in this workshop, they will get the update for the further program. And now we are about to complete this inaugural session. So I would like to invite Dr. Divya Mishra Madam for vote of thanks. Madam.
On behalf of Chemistry Department, I, Dr. Divya Mishra, would like to extend our heartfelt gratitude to each and every one of you who have or who will make this event a resounding success. I thank God Almighty for providing us the opportunity to organize this national workshop, and to our Honorable Vice Chancellor Madam, Registrar Sir, we express our deepest gratitude for your unwavering support and encouragement. Your leadership and vision have been a constant source of inspiration to us. I thank our subject experts, Professor Sanjeev Kumar Singh from Algappa University, for sharing his insightful keynote lecture, which included the etymology and the more than a decade-long drug discovery process. I thank Dr. Abhijit Kayal sir, Dr. Vinod Devarji from Schrodinger team, who will be sharing their expertise to train our participants. I thank our senior staff and our EMRC staff members for making us live. I thank all the students. We appreciate your enthusiasm and eagerness to learn. Your participation, I hope it will be truly inspiring. I thank all the esteemed participants for your active engagement and for your valuable contribution throughout the process. I hope your presence will enrich the discussions and feedbacks during this national workshop. Thank you, everyone.
Thank you, Dr. Divya Madam. Let us take a moment to pay tribute to our great nation. Let us show our respect and gratitude to our nation, its people, and its values. I invite all of you to rise and join for our national anthem. Thank you. Bharat Mata Ki. So with this, we are completing this inaugural session. Now, rest of the program will be carried out in LR 35 in old building. For MSc 2, you will have the lecture from Dr. Dilip Vasava sir and Dr. Divya madam. After this session, you will have the 2 minutes break.